Hi guys. Um, I'm just gonna give like this brief overview of the embryology of semester one. I'm gonna do it in two parts hopefully, and I'm sorry about the format as well. But if you have any issues or questions or anything, let me know. I'll message you or reply as fast as I can. So let's just get started. The female menstrual cycle and oogenesis. We've kind of gone over this in semester three as well, so it should be kind of fresh in your memory. But what I'm going to do is just kind of highlight the important parts and go over as much as I can. So, like we know, the hypothalamic pituitary gonadal axis is what really determines this whole menstrual cycle, right? So you have the relationship between the pulsatile release of the GnRH from the hypothalamus, and then from the hypothalamus it'll go into the pituitary gland. Still, this is found in the brain. Hopefully, I'm going to do it right. So the pituitary will contain will within this pituitary you'll have the release of these two hormones known as the FSH and LH. So FSH is the important part. Will go to your ovary. Will go to the follicle specifically, and you're able to sort of develop it as it goes on. And then hopefully the follicle will develop, and at the same time you'll have the oocyte developing, and essentially the idea is it'll give you a mature ovum at the very end, along with the LH, releasing that mature ovum to an extent, mature ovum as much as you can within the ovary, and then overall it'll go through the fimbriae and then into the sort of the ampulla. We'll just get to that. That's kind of the briefest overview. So yeah, that's pretty much it, and it's very dependent on hormones. You cannot have a menstrual cycle without hormones. So you have the hormones in question will actually allow for the maturation of the ovum and also the ovulation. But also, I did not mention this earlier, but you have other changes. The endometrium would change. The reproductive organs would change. And you would see sort of a cyclical change as well as mucus and cervical changes, everything because of these hormones. And body temperature will also show a variation, and you can use the body temperature to sort of detect ovulation and things like that. It's very important to monitor these changes, so you can sort of get a brief overview of what kind of stage of the menstrual cycle you are, maybe, and also help you plan for the future as well. So, typically, for infertility, you kind of have to assess these important aspects. So the hormonal levels, the changes in the ovary, the endometrium, and the female reproductive organs. And so if you monitor these cyclical changes, any abnormalities will help you sort of diagnose and discuss, is this person suffering from infertility or not? And also, they tend to be also assessed on their sexual development. I'll get to that in a bit. So, like we know, a menstrual cycle is a 28-day period, but this is not exactly 100% for everyone. It could be longer, it could be shorter. And ovulation will take place on day 14. And, we usually like to just say that the day one of menstrual cycle is actually the first day of menstruation. It's considered that way, so it's easy. To sort of as a reference. And now in terms of a clinical correlation, at this point you have amenorrhea. So amenorrhea is the absence of menarche. So menarche is, I believe, the first menstrual cycle of a female. So you have menarche in terms of puberty. If we're going to go back to the semester 3 physiology lecture, it was pubarche. And then you have telarche. I don't think this is going in order. Telarche, which is sort of the pubic hair. Telarche is the breast development. Pubarche is the pubic hair development, and then menarche is going to be the menstrual cycle development. So, yeah. Typically speaking, you could have abnormalities. It maybe not actually occur, and the menarche could not possibly occur, and this gives you the amenorrhea sort of thing. And it's because follicular maturation doesn't occur, and this can happen due to many reasons. Example, ovarian dysgenesis, so problem with the structure, and maybe hormonal defects as well. You have two types actually, so you can split this and you can say this is primary, and there's also another one known as secondary. So primary means that there's absolutely no menarche at all. You have not reached menarche, you have never gotten a period. And you can classify this if the secondary sexual characteristics haven't developed by age 13, and if the menarche hasn't occurred 5 years after initial breast development, or if the patient is 15 years or older. Things like that can help you identify, okay, so is this primary amenorrhea or not? And then secondary amenorrhea is sort of, you've got your menarche, you've got your periods typically, but after some time your periods will cease. Like, they will just not come. And if it doesn't come for more than 6 months, you can classify this as secondary amenorrhea. Polycystic ovarian syndrome is very, very prevalent in this day and age, and it's kind of like these presence of small cysts within the ovary, and these small cysts will be fluid filled, right? And these are fluid filled sacs. These are what cysts are, and it causes the ovaries to become enlarged. Yeah, and typically speaking, the cysts are actually just follicles that haven't released, and this can happen due to a multitude of reasons. It could be due to a hormonal issue, and many other issues. I'm not really sure about the exact examples, but so overall, you have these little cysts within the ovaries, and these cysts are basically follicles that haven't released. And why have these follicles not released? It could be due to hormonal disorder. So maybe it's not responding well, maybe there is not enough hormones, things like that. But anyways, typically people who have polycystic ovarian syndrome can typically have irregular periods, and possibly less frequent and less commonly ovulation. So what they're saying is the periods themselves don't come in a regular fashion, and when they do, you may or may not even have ovulation. So it is possible to have anovulatory periods. So you would also have excess androgen levels, and this causes, physical characteristics such as excess facial hair or body hair, or even acne can be seen as well. So that's pretty much it. And then in terms of the ovulation and the ovum transport, we kind of know the first part, 14 day of a 28-day cycle. And when ovulation actually occurs, you have the follicle, it ruptures, releasing the oocyte, the ovum, and then it passes through, due to the fimbriae, it passes through, and it's aided by a couple of things. So the ultimate destination is to the ampulla, right? So this is the ampulla, and the sperm actually comes the other way around. So here, and hopefully they meet, they fertilize, and then they go to the fundus of the uterus and then implant. Sorry, not fundus of the uterus, the body of the uterus, specifically the posterior wall, but that's not important. What is this? Okay. At the moment, it's not. I'm going to get to that in a bit. And so the transportation to the ampulla, it's aided by mucus and secretions within the fallopian tube, another word for the uterine tubes, and the ciliary action. And so there's lots of cilia, and they help move the ovum across, and also the actions of the muscles themselves within the fallopian tube. So like I mentioned, they help with the direction of the movement of the ovum, but also of the spermatozoa during fertilization. Now, oogenesis is the whole situation known as gametogenesis. So if I were to talk about gametogenesis, it's just talking about the production of gametes. So you have the male one and you have the female one. So the male one is known as the sperm cell, and this one is known as the egg cell, or also ovum. Yeah. So where do these come from, though? These are gametes, right? This is a gamete, but where do they come from? Now, the predecessor, the whole root of this, the whole sort of source of this is PGCs, also known as primordial. It's best, instead of using the abbreviation, to sort of mention this in the beginning, primordial germ cells. So primordial germ cells give rise to gametes through a process known as, either in the females, oogenesis, or for males, spermatogenesis. So it kind of goes through a couple of other steps, and then it is able to go to gametes. So anyways, we have the oogonium. I'm sorry if I'm not pronouncing it right. But you have the oogonium, and this comes from the PGCs, right? The progenitor cells. And there's a little story related to the progenitor germ cells. So, sorry. Primordial germ cells. So the primordial germ cells, they actually arise from the sorry, I kind of blanked out for a second. So they arise from the epiblast in the second week. I'll get to the point with the epiblast and everything a little bit later. And then, from the epiblast at the second week, they move towards the primitive streak. This is in the third week. And then by the fourth week, they would have moved to the wall of the yolk sac. And then from the wall of the yolk sac, they would have migrated to the respective gonad. So in this case, it would be the ovarian cortex. So anyways, during the migration process, actually, these primordial germ cells, they will mitotically proliferate. And this mitotic proliferation, because they have nothing to do during its journey, it decides to just mitotically proliferate. And then this gives rise to oogonia, also known as the primordial ova. And then each primordial ovum becomes a primary oocyte. And this is also a couple of, differentiation ready for meiosis, I think is what it means. It gives rise to the primary oocyte. And actually, the primary oocyte doesn't automatically go down to the secondary oocyte, unlike spermatogenesis. It's actually arrested. And specifically, it's arrested at the first meiotic prophase. So you have meiosis 1 and you have meiosis 2. So in meiosis 1, you have prophase, metaphase, anaphase, and telophase. But specifically prophase, it actually has 5 steps, okay? So fourth step out of 5. So the fourth, just before the fifth step, is known as the diplotin stage. And this is what it is here. So it's the diplotin stage, and this is where it's arrested in the primary oocyte. And prophase itself, among other things, it involves things like recombination and recombination is very important for genetic variation. Anyways, that's it. So the primary oocyte is just there. It's just kept inside this follicle. All these follicular cells kind of surrounded, forming this follicle around it, and giving it the name primordial follicle. So primordial follicle is what it's going to be way until puberty. So until puberty, even after birth, it will remain this way until puberty, until it becomes a primary follicle, and then from there it'll become secondary follicle, an antral, early stage, late stage, and then a Graafian follicle, and etc. So so this is the type of cells that are actually making up the follicular cells. They're epithelioid cells. So flat epithelioid cells. And like I mentioned, it'll be known as a primordial follicle. And the ovary at birth, it would contain around 400,000 primordial follicles. And again, it sort of involves the primary oocytes that are arrested at meiosis 1. Arresting sort of happens due to a lot of chemicals. A couple of chemicals and the chemical signalings and here and there. So they end up just being arrested. And at puberty, so this is actually talking about now the maturation of the follicle. So it's not like going in a specific kind of order. So now we're just going straight through gametogenesis, right? So including the follicular development. So the primordial follicle, so you have the primordial follicle, it sort of becomes a primary follicle. So the real difference between the primordial follicle and the primary follicle is that, first of all, you have the epithelioid cells. So how I mentioned earlier, these flat epithelioid cells will now become cuboidal in shape, and also they will become more stratified. So the stratification being more layered, actually, and also the change in the sort of the shape per se will give the sort of a different label now. It's known as a primary follicle, and they're actually known as no longer follicular cells, but actually by granulosa cells. Granulosa layer, granulosa cells. This one is special. This layer is now special because it's able to give you something known as a zona pellucida, which I'll get to in a bit. So like I mentioned earlier, the primary oocyte is kind of like here, and then it's kind of like the granulosa cells, and then in the middle you have the glycoprotein layer. Something I kind of highlighted here, I just wanted to mention this. There is a stage, so there are two stages. You have gonadotropin, I'm just put gonadotropin hormone dependent, and then you have gonadotropin hormone independent. So actually, the stage where you have the primordial follicle turning into the primary follicle and also the secondary follicle, this one is actually known as gonadotropin independent. So actually, it doesn't really depend on the gonadotropin hormones. So it's only like the antral follicle. So like I mentioned here, primordial to primary to pre-antral stage. So at this point, this is known as gonadotropin independent. But actually, after the antral stage, so post-antral stage, all the way till the Graafian follicle, I believe, this is actually dependent on the gonadotropin hormone. So that's just to, in case they ask for like an MCQ and everything, just mention that the primary oocyte stage, so the primary follicle stage, the primordial follicle stage, those kinds of stages, they are gonadotropin independent. So that's what I wanted to talk about. Yeah. I believe in this phrase, the sentence right here, they're mentioning like after ovulation, you have the rupturing of the follicle, right? So the the ovum is able to pass through, but then what happens to this follicle that has been ruptured? It actually doesn't just stay like that. It actually turns into something called the corpus luteum. So it's kind of like this yellow flower looking thing. And it actually helps with maintaining the lining of the endometrium. And this is actually known as the corpus luteum, and it helps release progesterone. And if fertilization doesn't occur, it'll turn into corpus albicans and just atrophize and just, fade away. Typically, like in terms of a clinical correlation, it's just the failure to mature, to produce a mature ovum. So like I mentioned earlier for the PCOS, you can have anovulation. So anovulation is where the ovaries don't actually release a mature ovum during the menstrual cycle. So it's actually, obviously would mean that the lady's infertile. And it could be due to many reasons. It could be sex hormone production is affected, the ovarian structure and function are defected, and also, you could have menstrual cycles because the actual endometrium would still be affected by the hormones, right? So the whole thing would be there, the whole building up and everything. But the thing is, it's just not going to contain that ovum that's going to be expelled as well. It's not going to have that. So that's why it's known as an anovulatory cycle. So basically, what determines a female fertility is the follicle number and also the responsiveness to sex hormones in each menstrual cycle. That's pretty much what I was trying to mention there. But naturally, you can also have anovulatory cycles. It doesn't have to mean that's abnormal all the time. You can have, like, after the first period, after menarche, a few cycles after menarche, and also childbirth, you can typically have anovulatory cycles, and this is completely normal. But also, you can have it when you have, like, hormonal treatment, like the birth control pills. You can also have anovulatory cycles because these birth control pills purposely hinder your ability to have like follicular maturation. And so that is why it's called birth control, because you don't have that mature egg that's released. And so that means that it's not going to be fertilized. And so you won't have a baby. That's pretty much what it means. Yeah, it's like I mentioned earlier, the PCOS. And in anovulation, people experiencing this, the cycle length may vary, and also you'll typically have irregular cycles, which is very inconvenient. Okay, so now we're going to go into spermatogenesis. I just wanted to mention a couple of things. It is a bit different compared to oogenesis. This whole process is a continuous thing. But the female one is not a continuous thing. You have arresting at certain stages, right? So you have arresting at meiosis 1 at the first stage, and then meiosis 2 when it is a secondary oocyte. So when it is a secondary oocyte, the meiosis 2 at, I think, metaphase 2, it is arrested at this stage. And so until it is fertilized, it doesn't finish its complete meiotic phase. So that's there. And also another thing I wanted to mention. So first thing is that it's not continuous. The continuity is a difference. And also another thing is that you have this stage called spermiogenesis. So if we were going to be completely like simple here, gametogenesis, the whole meiotic situation, would happen until the formation of spermatids. So after the formation of spermatids, it doesn't look like the typical sperm that we know, right? So it doesn't have that shape. It has to undergo a process called spermiogenesis. Within the seminiferous tubules, that's actually the location where spermatogenesis occurs. Spermiogenesis, the walls of it. So anyways, this is a process where just the development of the cell happens. So you have a difference in the cytoplasm, more of the cytoplasm is shed actually, and you have this special stuff, the tail, the mid-piece. Sorry, I didn't draw it properly. Anyways, there's a better picture over here. So the head, the neck, the mid-piece, and the tail. And so all of this will be developed during spermiogenesis. So that's the second thing. So you have that extra step known as spermiogenesis to give you that proper shape, that proper function, therefore the proper ability to actually fertilize. And the third thing that what I can think of is the timing. So the timing is different. So actually this comes from the primordial germ cells, right? So the primordial germ cells are there within the male reproductive system after birth as well, all the way till puberty as well. And then, from the start of puberty, you just have these primordial germ cells going under the process of gametogenesis to produce the gametes. So this whole process happens after puberty, like at the start of puberty. That is what kickstarts it all. But for females, it's a bit different. For females, it starts, so at the embryonic development, you have, and the embryonic development, you have a set number of PGCs will give rise to primary oocytes. So these primary oocytes will be arrested, and then from birth till puberty, you don't have anything. It remains the same way. But it's only at puberty you have a select few, and typically once per month you will have one mature ovum release. So the timing is very different for both of them. That's pretty much what I wanted to mention. Yeah. Hold on, let me just... Okay. So that's pretty much it. So the overall kind of thing that you will get is a spermatozoa, a sperm cell. That's the overall goal of spermatogenesis. Like I mentioned, seminiferous tubules of the testes, and yeah. In terms of clinical correlations, spermatogenesis and oogenesis, so gametogenesis, really impact male and female fertility, respectively. So fertility is a crucial selection factor, and in general, when you compare it to the other species of animals, humans tend to have a remarkably poor fertility. So that could be an MCQ, I think. So in terms of semen analysis, you have to think about the sperm concentration, so the sperm count, and also motility and morphology, and volume, chemical analysis, liquefaction time, immune reactions is also checked. And there are also normal ranges, which I think is more... mentioned, I think, in semester 3 physiology and reproductive thing, I think, again. So better to check that out. Yeah, and these ones just mentioning the normal values in this one as well. Sorry, I didn't do that in red. And there are clinical correlations when you have an abnormal count. So when you have a low count, it's known as oligospermia. So low count is known as oligospermia. And then when you have no sperms at all, that's called azoospermia. Again, I'm not sure about the pronunciation. Low sperm count may be due to many reasons. For example, could be due to problems in sex determination and diffraction, and this can lead to structure and functional anomalies within the reproductive system. So Sertoli cells, I did not mention this, and the lecture note didn't really mention it, I think, but Sertoli cells are extremely, extremely important for the development of the sperm. So without Sertoli cells, you won't exactly have functioning sperm. So again, number of Sertoli cells determine the sperm count because they go hand in hand with the development of the sperm. And then also hormonal problems, and also time since last ejaculation is also a factor. Now the actual gametogenesis part is relatively finished, and I'll be moving on to the prenatal development. Okay, so the prenatal development is basically from a singular cell, like from the zygote. How would you get it turned into a multicellular human being infant upon birth? So that kind of situation, this kind of prenatal development is split into two periods, so the embryonic period and the fetal period. So the fetal period isn't really mentioned much in the lecture note. It's mainly going into depth within the embryonic period. And the embryonic period just is like around 8 weeks after fertilization or ovulation. And obviously fertilization takes place after ovulation. And this is like a sort of a graph, if you will, like a sort of a picture just depicting what I mentioned. So within the first 8 weeks, fertilization or conception, these first 8 weeks you can sort of talk about this period as being the embryonic period, and then after that it's just known as the fetal period, and then after being birthed it's different. So yeah, that's pretty much it. And then a clinical sort of situation that we're probably quite familiar with, to assess the days of pregnancy or period of gestation, which is the same thing. You have to first ask about the ovulatory age, post-ovulatory age. So when was the last time since their last regular menstrual period? Because they meant regular because you can't really assess if someone has irregular periods. So this method would not work if someone had irregular periods. And also symphysis of pubis, fundal height, and ultrasound scanning. These ones would sort of go hand in hand with this one. So using regular data that they have accumulated over time, they can compare what is to be expected at that certain stage, at that certain time, at that certain age, and then they're going to use this to identify, okay, so this fetus is sort of this many weeks, you're this many weeks into pregnancy, and things like that. So yeah, that's pretty much it. And in terms of the embryonic period, we mentioned that it's 8 weeks, and it has roughly around 23 stages after ovulation, so after fertilization. So it includes fertilization, and the first stage is fertilization, and it starts with fertilization, and what it means is you have the fusion of male and female germ cells, and so these germ cells give rise to the actual nuclei, and these nuclei, you specifically have to mention that it's pronuclei, and when these pronuclei fuse, that's when you get the zygote. And where does it happen? It happens in the ampullary region. Remember how I mentioned that from the fimbriae, due to the cilia, due to the muscle contractions and the secretions and whatnot, you have the ovum reaching the ampulla, and it waits there and maybe probably calls the sperm through chemical signaling as well. The sperm also reaches the ampulla, and then fertilization occurs, and then they will sort of go roughly the phase next would be implantation, the biggest stage. But there are many stages in between, so I will talk about that too. Fertilization occurs within 24 hours of ovulation because the egg cell has a lifespan of only 24 hours. So roughly 24 hours. So when talking about fertilization, we have to mention the two important sort of phases that the sperm has to go through in order to ultimately be able to fertilize the egg. So to reach the egg, first of all. So capacitation happens first. So this is like the first, upon entering the female reproductive tract. Upon entering the female reproductive tract, the sperm will undergo a series of changes. So these physiological changes involve increasing the motility of the sperm and destabilizes its cell membrane, all to just make sure it reaches the egg as fast as possible. So this situation occurs due to uterine fluids and other sort of situations within the reproductive tract. So that's why female reproductive tract. So this is what causes and triggers capacitation. It takes a while, but once it happens, it's able to reach the egg. And just to sort of, I'm gonna talk about this in a bit as well. About this one in a bit. But basically, when the when the first sperm, when it reaches the egg, it has to go through layers, right? So the layers include cumulus oophoris, corona radiata, and the zona pellucida. So 1, 2, and 3, and at the zona pellucida, it binds to receptors, and this triggers the second stage that I mentioned. So the second thing that happens to the sperm is the acrosomal reaction. So the acrosomal reaction occurs when they bind to the receptors, and this triggers a release of enzymes on the sperm head. And these enzymes include, but is not limited to, trypsin and acrosin. And you have this membrane containing all the enzymes. They burst, and these enzymes that I mentioned release, and they go through the zona pellucida and sort of degrade it in order for the sperm to actually enter through to the egg. Yeah, and then upon gaining access to the cell membrane of the ovum, and this sort of links it to this now here. You remember how I mentioned that it was a secondary oocyte, the mature ovum that's released from the follicle, the Graafian follicle initially. So the secondary oocyte is arrested at metaphase 2, right? So upon the access of the cell membrane of the ovum by the sperm, the meiosis is completed. So meiosis is completed, and now you have the proper mature ovum. It's a complete mature ovum. So gametogenesis is over. And also I wanted to mention another key difference between oogenesis and spermatogenesis, I kind of forgot, is the fact that you have no polar bodies in males and you have polar bodies released in females. So within the male reproductive thing, you have basically 4 spermatids formed, right? But actually for the female, you only have 1 mature ovum formed, which is really weird. But when you think about it, it's actually the first meiosis releases 2 cells, right? But the thing is, it's the primary. It's a secondary oocyte, isn't it? Yeah, it's a secondary oocyte. It forms the first meiosis, will give you the secondary oocyte, and then the first polar body, and then you have the first polar body here, and then over here the second polar body is formed at the second meiosis. So the polar bodies in question, they're not really that significant in terms of the function, but it's better to actually show that you are aware that the polar body is released. So that's why I highlighted it. And there are certain reactions within the ovum that prevents polyspermy. So polyspermy is the action of many sperms entering through and sort of trying to fuse with the nucleus, which will give you a completely disastrous ending. It's not going to have a zygote anymore. It's no longer going to be diploid. So there's going to be a lot of structural abnormalities and the genetics part of things. You can see in the other notes. So the 2 main mechanisms that prevent polyspermy is the removal of receptors on the zona pellucida. So therefore, if the sperm is able to, enter through the different layers, corona radiata, cumulus oophoris, they won't be able to pass through, they won't be able to bind to the receptors on the zona pellucida because there aren't any. And also, the polarity of the cell as well, the ovum, the egg cell, the polarity is also changed. I don't know why it's not mentioned, but the polarity is also not there. Polarity will be changed, and so that also prevents polyspermy. You can see here's just a picture, and upon fertilization you have three things that will happen. So obviously the diploid chromosomes will be there, so the zygote will be diploid, not haploid, and the chromosomal sex is determined too, and that is actually determined by the sperm. And cleavage is the next stage, the stage number 2. But first of all, I'll mention I mentioned about the corpus luteum, right? So the corpus luteum, if fertilization occurs and implantation occurs, it'll actually exist until the 4th month of gestation, and then after that you will have the placenta being formed, and then that will take over the action of the corpus luteum in that it releases progesterone. In terms of clinical correlations, you have IVF. So IVF is an assisted reproductive technique where it combats the inability of a couple to actually, a couple that has like problems with fertilization. This could be due to a structural or hormonal, again, a problem. It could be with erection, it could be with poor sperm count or quality, etc. So this happens when the ovum of the mother and the sperm from the father are taken and then mixed outside the body, and then the zygote that arises from this will be injected back into the mother, and sorry, it's like the embryo, actually. It's not the zygote. It's like the embryo is then cultured and then transferred into the mother. So now we're going to talk about the second stage, is cleavage. And cleavage starts just after fertilization. So you have the zygote, and it's just one cell, right? So now you have multiple mitotic divisions, but this is a bit different from a normal cell in that it doesn't actually have a normal growth cycle. It doesn't have that typical phase. So you don't have the interphase, you don't have the other phases. So you don't actually have time for growth of the actual cell. So actually what you have is it gets smaller and smaller with each division, the cells within that are formed from the mitotic division of the zygote. So so it divides mitotically, and it forms a morula. So the morula, it consists, it's like these ball of cells. It's always described as a ball of cells. And these morula, think of like a bunch of grapes. So you have a bunch of grapes here, but each individual ball is known as a grape, right? So in this case, a morula is a ball of cells, and each cell is actually known as a blastomere. So yeah, this is what I mentioned earlier. And a thing about these blastomeres is that they're spherical. I didn't really draw it well, but it's spherical, and they touch each other, but there are no specific specialized intercellular junctions. It's not there. It's just loosely touching, and also there are no extracellular matrix. The extracellular matrix is not there. And now in terms, I would say this could be classified as a clinical correlation in that it's used for stem cell research and stem cell therapy and everything. So these individual blastomeres, they are separated and can be used as embryonic stem cells, and it's totipotent, meaning it has the absolute ability to differentiate into anything that it desires. You desire. Anyways. So the morula with 16 cells will end up becoming a blastocyst eventually. This is because within the cell itself, you'll sort of have a kind of cavity. So this cavity is known as a blastocystic cavity, and it sort of arises between the individual blastomeres. This cavity just happens to come over time because of the uterine fluid, etc. So it's not really that important, but there is a cavity, and it's called a blastocystic cavity. And this is what gives the name now. No more morula, it's actually a blastocyst. Now, the next stage after the formation of the blastocyst is the hatching. So it's a hatching. It's called the hatching. stage, it's basically where the blastocyst, and it's still with the zona pellucida, right? Think about it with the egg cell. It gives it actually is the cell component of the zygote, right? Like the sperm just provides the nucleus and all the little other things, but overall the ovum provides the organelles. It provides the actual space, cellular structure, pretty much, but it also contains the zona pellucida. It hasn't disappeared for now. But at the third stage, it's hatching, and which is called hatching, is basically where the blastocyst frees itself from the zona pellucida, and it's pretty much shed, it's gone, and then the blastocyst is able to increase in size now. It's able to enlarge, but I'll get to that in a bit. At this point, when it's hatching, when the zona pellucida is shed, it's around 4 to 5 days, and the size is 0 to 1 to 0.2 millimeters in diameter. And at this point, I just wanted to talk about the structure. So this is an outer cell layer. Okay, so let's think about this ring for now. This ring you see here is known as the outer cell layer. It's known as the trophoblast. So the trophoblast is important because it gives rise to the placenta, which I'll get to in a bit, and the inner cell mass here. So the inner cell mass, it gives rise to the embryonic embryo proper, and it's called the embryoblast. Embryoblast. So just another word to the same thing, pretty much. So this inner cell mass gives rise to the bone embryo proper, and these are known as pluripotent cells now, no longer totipotent. It's pluripotent, which is similar to totipotency, where it basically has the ability to virtually turn into any cell, but it has limitations. So there are slight limitations. Now, anyways, like I mentioned, the ampullary site is still within the ampulla, so it's just making its way down to the implantation site. And just a small clinical correlation, a blastocyst development will get arrested at around this stage, maybe, if there are any genetic abnormalities. So stage 4 is known as implantation. Now this happens like around 6 days after fertilization, approximately. And like I mentioned earlier, after hatching, so it's no longer constricted with the zona pellucida. It now has the ability to grow even bigger. So it enlarges, and now there are tight junctions forming between the trophoblastic cells. And the migration of so basically makes its way to the site of implantation, and that is specifically, if we were going to talk about the structure, it's here actually. It will be in the posterior wall of the body of the uterus near the fundus. So the fundus is at the top, right? So the fundus is at the top, and then you have the body here, typically, roughly, and then the posterior wall of the body, just at the fundus, is where, just near the fundus, is where implantation occurs. So that's one thing to remember, and also adhering to the endometrium, which has to be implantation which occurs at the endometrium. The endometrium itself has to be quite ready to receive the embryo. At this point, it's the blastocyst. Sorry, so it's has to be ready, and this window occurs for like around 4 days. And the inner cell mass, when it tries to implant, like this point here, this implantation, there are, it's actually kind of like a little bit more than just implantation. It sort of involves apposition. So apposition is where it aligns itself to, in order for the correct position. It's like when you're kind of hugging someone, when you're hugging it like face to face, you have to face each other, right? Or else it's a bit awkward. And this is similar to how the inner cell mass is aligned. So the inner cell mass, like I mentioned, I forgot to mention, inner cell mass is going to be the part over here, right? So this is going to be the inner cell mass. Oh yeah, the embryoblast. So this point will be aligned facing the endometrium. So it's like they're hugging each other, and so it has to align, and so that's why it's known as apposition. And then it attaches, so the receptor sort of situation, it happens, and then invasion happens. So invasion is where it actually invades through the endometrial tissue, or this maternal decidua, and sort of tries to get itself inside, and then the other stages will happen after. So it's gonna, so it disrupts and invades the maternal tissue, and this sort of helps to end up enveloping the blastocyst within all this maternal decidua. So yeah. A clinical correlation would be if you had implantation occurring that is outside the typical location that I mentioned earlier. So this is the normal location. If it were to implant itself somewhere else, that would give it the term an ectopic pregnancy. And basically it can happen in any of these situations. So it can happen in the intramural area, so over here within the uterine body itself here. It can happen in the ovarian area here. It can happen in the isthmus, which is actually the narrowest part of the fallopian tube. That would be quite bad. Fimbriae as well is quite bad. And ampulla, it is possible, but it could lead to damage to the actual uterus in the future if this were to actually be continued, like if the pregnancy were continued. And also it can happen over here, so the corneal kind of area, the sort of areas where you get the corns here. That's also possible. And cervical, and I think abdominal, which is quite, that's a thing too. Now at this point, at this stage, when after invasion occurs, you have during invasion, like during the process of invasion, actually, you have the development of the bilaminar disc. So this is extremely, extremely important. So remember how I mentioned that the inner cell mass is facing the endometrium, right? So it's going here. The endometrium is actually, it's here, this white area, so it's facing this way. So they're facing each other at some point, yeah? So the inner cell mass is now not going to stay with the same name. It's going to be a little bit more varied. It's going to be a bit more different. It's going to change. It's going to split into two layers. So the first layer is known as the epiblast. So let's just say that I'm going to split this line, and this is known as the epiblast. So epiblast is also known as the external, it's external, and it's known as epiblast. And I also like to remember as endometrium because it's facing the endometrium the most, so it's actually closest, right, to the endometrium compared to this layer. So this one is going to be known as the epiblast. E for epiblast, E for external, E for endometrium. And the layer below it is called the hypoblast. So the hypoblast is actually facing the blastocystic cavity. Like I mentioned earlier, for the blastocyst, it actually has like this fluid-filled cavity, right? So the hypoblast is actually the one that's closest to this cavity here. So that's quite important. So it's like important in the sense that it splits into two layers. And also, like I mentioned earlier, that outer cell layer that was here, and you have the inner cell mass here and the outer cell layer, right? The outer cell layer is known as the trophoblast. It's not going to stay as a trophoblast forever. It's actually going to split as well. Split in the sense it's going to have the syncytial trophoblast around here, and it's going to have the cytotrophoblast around here. So the cytotrophoblast is kind of like here, and so the outermost area would be the syncytial trophoblast. So you can see over here, and then you can see over here. This is the cytotrophoblast, and then this is the syncytial trophoblast here. So the syncytial trophoblast is special. Hold on, I'll get to that in a bit. It's important to remember that the bilaminar disc is slightly oval in shape. In case they mention the shape or anything, it's just better to know. And the epiblast and the hypoblast have another difference, not just the location, but also in terms of the histology. You have the epiblast consisting of high columnar epithelia, and then the hypoblast, which is actually composed of like cuboidal or polyhedral cells. So that's important. And overall, the bilaminar disc is quite a flat disc. So the bilaminar disc, just to recap, is composed of the epiblast and the hypoblast. And there are something known as amniogenic cells. I'll get to this in a bit as well, but amniogenic cells are basically cells that contribute to the amniotic cavity. So if you were to see a picture of this developing structure at around, you can also call this a conceptus, what is formed after fertilization. That's what it was in the book anyway. But you kind of have this cavity that's formed eventually. This being the epiblast layer, this being the trophoblast, or sort of like the syncytiotrophoblasts and everything. So the outer cell layer, let's just keep it as simple as it is. So the outer cell layer, yeah, and then you have the epiblast, and then you have the hypoblast. So the epiblast, and between the trophoblast and the epiblast, you have something known as the amniotic cavity being formed, and this is initially formed by amniogenic cells. Yeah, that's pretty much it. So that's that, I believe, and it's this kind of picture, if I'm understanding correctly, like it would be kind of like this, and if, let's say, this was the epiblast and if this was the hypoblast, and this is the amniotic cavity, and if I were to cut this, I think this is sort of like if I were to cut it in like a 3-dimensional way, like this way, and then I see like a cross section like this is pretty much what it would be, something like that. And now, like, we're within the implantation phase, right? So we've passed through invasion, so now it's actually trying to implant itself snugly inside the endometrium. And like I mentioned earlier, the trophoblast gives you a syncytiotrophblast and a cytotrophblast. So this is a thin inner layer, while the syncytiotrophblast is huge. It's quite like weird looking. It's like jelly, if you will, and it's composed of many cells, but it's not like a definitive specific. You can't really say that these are individual cells. It's actually, the boundaries of these cells have actually disappeared, and now you kind of just have this giant mass of lots of nuclei of these cells earlier. So now they're just nuclei. So this is a protoplasmic mass. I think that's how they describe it. So when the whole embryo is embedded in the endometrium, the syncytiotrophblast will come into contact. So the syncytiotrophblast is actually, in terms of the function, it's what helps invade into the maternal blood. So this one has enzymes and all sorts of things. It's able to break the maternal decidua, the maternal tissue down, and it helps sort of settle the entire structure inside the maternal endometrium. Yeah, the entire embryo. And when it's completely when it's in the process of being embedded, the syncytiotrophblast, while it's degrading through the maternal decidua, it means it'll also come into contact with maternal blood, so these maternal blood vessels. And so when they come into contact with them, they break it down as well. And so they end up forming these structures known as chorionic villi. Now, that is what I meant by trophoblast being able to contributing to the formation of placenta. Because through the syncytiotrophblast, through this interaction with all the maternal blood and everything, you have this chorionic villi being formed, and this is sort of like the initiation of the formation of the placenta. And you can also mention that this is where the uteroplacental circulation is sort of starting to be set up. As the blastocyst burrows into the maternal decidua, the area is actually now going to be sealed with a fibrin plug. So this is now, it's like a sort of healing process for the maternal endometrium. Yeah. And this is like, typically it happens around day 9, right? This happens like around day 9. And this is the sentence that I mentioned earlier, up above. It's like this cavity, the amniotic cavity, occurs just above the epiblast. So yeah, so around here, this is like what they're mentioning, like the amniotic cavity. And within the syncytiotrophblast, I believe they're mentioning this part, you have these irregular kind of vacuoles being formed, and then they fuse to form lacunae. So lacunae from vacuoles, they fuse to form lacunae within the cytoplasm of the syncytial trophoblast, and then at the end you kind of have sorry, not the end, as in like this specifically at this end here, you have hypoblasts. Here within this area you have hypoblasts. So remember, you have this layer, bilaminar disc. The epiblast is linked to the amniotic cavity, right? So we kind of want to give the same treatment to the hypoblast. We can't give them the same amniotic cavity. That's just not right. Instead, you can settle for another cavity, and this is known as the exocoelomic cavity. Again, I'm not really sure about the pronunciation, but this arises from exocoelomic membrane, which is it comes from cells, probably according to the book Langmann's, it could come probably from the hypoblast. So these cells make up this kind of membrane, and then this membrane will form this cavity. Yeah, so this cavity is also known not only as the exocoelomic cavity, but also the primitive yolk sac, which we have heard a lot about. So this is the primitive yolk sac, very, very important. And just for reference, it's like it gives you the extra embryonic mesoderm, which we will see in days around 10 and 11 and 12. For now, I will kind of pause, and I will make another video talking about the other days and the other parts of embryology.